Okay, what we've been talking about so far is cellular respiration in the presence of oxygen. Let's talk briefly a second about what happens in the absence of oxygen. And I'm going to use the book website for this more and just draw on the slides and see how this works. This is kind of an experiment anyway. Okay, normally if you have oxygen present, glucose is broken down, goes through glycolysis, is broken down to pyruvate. Pyruvate goes into the mitochondrion and does the rest. If there is no oxygen present, one of two things happens. In a yeast cell or other organisms, pyruvate goes through a different set of chemical reactions. There's no oxygen present, so it doesn't go into the mitochondrion. The NADH, the energy from that is used to convert pyruvate essentially into ethanol. Ethanol is a form of alcohol. Notice CO2 is a byproduct, so we put our yeast in a solution with some sugar, and it produces carbon dioxide if we want to make bread with a very little bit of alcohol. Or if we want to make wine, it produces a little carbon dioxide and a lot of alcohol. In our muscles, we're using our muscles faster than our lungs can keep up. We make pyruvate. We use the energy of NADH to convert that pyruvate to lactate, also known as lactic acid. So the general overview of this is, let me go down to this slide, that in your cells, glucose goes into the cell, into the cytoplasm. It's converted to pyruvate. If there's oxygen present, cell respiration. If there's not oxygen present, in yeast, ethanol, in us, lactate. Okay? One big thing, we only get 2 ATPs out of this. Here we get 38. 19 times less energy. Therefore, lactate buildup makes you fatigue quicker. And we can come back to that and talk a little more about that at a different time. Okay. So, we have fermentation and pyruvate. Now, let's take a look at this overall. What about other things? Well, we can not only break down glucose and carbohydrates for energy, that's our normal pathway we just talked about. We can also break down fats for energy. In fact, we use fats for energy a lot. The glycerol part, remember, fats are made of glycerol and fatty acids. The glycerol part enters right in the middle of glycolysis and is broken down to pyruvate and goes that way. The fatty acids can be converted to acetyl-CoA into the citric acid cycle used to make ATP. Numbers of ATP don't matter. Proteins, on the other hand, broken down to their amino acids, they can either enter the cycle in one of 3 spots, depending on what kind of amino acid it is. Not that important to us, but notice, when you break down proteins, one of the byproducts is a very toxic ammonia compound. We use that we turn that into urea, into urine, and we excrete it in our urine. Okay? So we can use proteins, carbohydrates, and fats in the same method. It's not just sugar that we have to use to break down for energy. And remember, if there's no oxygen, all of these go to fermentation. If no oxygen, all go to fermentation. The last slide is, how does our body, quote, know? do we know to make energy or not? Well, it just so happens that one of the enzymes in glycolysis is called phosphofructokinase. Sweet name. That is an allosteric enzyme. That allosteric enzyme is inhibited by ATP. So if we have plenty of ATP, we have made plenty of ATP. ATP inhibits, here's our phosphofructokinase, here's its allosteric site. ATP binds onto there and warps the enzyme so it doesn't work. It's an allosteric inhibitor. So now we don't get glycolysis. What do? We take any extra stuff and store it away in different products. Fats, glycogen, something else. Citrate in the citric acid cycle. If lots of pyruvate's going in here and the cycle isn't quite keeping up, so you're getting an overproduction of citrate, that also inhibits it. AMP, which is an A with one P, means we've broken off two from, this is ATP, tri. Sometimes we break off two. That stimulates phosphofructokinase. It's an allosteric exciter, if you will. So, let's review. So here's fermentation again. And let's ask this. How does our body, again, how do we know to make lactic acid? Well, it's pretty simple. As you make pyruvate, and as your body's doing glycolysis and you're burning energy, you're breathing oxygen. Remember, all the way at the end of the electron transport chain, let me go back to the end of the electron, well, let's look at this a minute. NADH starts to build up. And I'll show you why it starts to build up in a second. It starts to build up, and so as it builds up, some of these pyruvates, they can't diffuse into the mitochondria because we're getting a backup. So they're stuck out in the cytoplasm, and we're getting a buildup of NADH, so this starts happening. Because remember, in the electron transport chain, all these NADHs that are made from breaking down glucose are dropping off their electrons and they're moving along, and oxygen is used to remove those electrons from the chain. If we don't have enough oxygen, we get a backup in the chain. Kind of like a backup if no one is driving the car off the assembly line. You get a backup in the chain so NADH can't drop its electrons off. So you get a buildup of NADH. So now you're getting a buildup of NADH, you're getting a buildup of pyruvate. Boom. Break it down to lactic acid.